This is Biology 101, Chapter 12, the cell cycle. Okay, so in the first two chapters of this module, we talked first about the cell and cell structures, all of the various parts of a cell and how they work together for the cell to function. And then we talked about the plasma membrane and specifically transport across that membrane to look at how the cell gets materials in and out. So those two parts of this module are very much focused on the cell's daily activities, how it performs its daily functions, and how it survives, really. But if we remember, one of the key aspects of the cell theory is that all cells come from pre-existing cells, and we know that all living things are going to have the ability to reproduce, and cells are the most fundamental unit of life. And so we're going to now spend the last two chapters looking at how certain types of cells go through this process of cell reproduction, and we call that cell division. Now, in a cell division process, we end up with two genetically identical daughter cells, and this is because the original parent cell will grow to the point where it's very large, and it contains enough material inside that it can now divide in half, and each of the two resulting cells will look identical to the parent cell. The thing that has to happen is that the genome, or all of the DNA inside of the nucleus, has to first double before the cell divides. And that's really where we're going to focus much of our attention in today's discussion. So mitosis is a part of the cell division process in which the genetic material in the nucleus is divided. So mitosis is really just a nucleus division or a nuclear division, whereas a cell division is going to be something that occurs in the later stages of the cell cycle called cytokinesis. Now, we're going to look in these two chapters at two different types of cell division. Mitosis is the type of nuclear division that we see in somatic cells, and meiosis is the type of nuclear division that we see in gamete cells. And these processes are going to be similar but also very different. So somatic cells are going to be all the cells in your body except for your gamete cells. Gamete cells specifically are your reproductive cells. So typically we think of in humans your egg and sperm cells. The differences between these is that gametes are going to be haploid cells. And what that means is they only have one copy of every chromosome. Whereas in all the rest of the cells of your body, in your somatic cells, those cells are going to be diploid, meaning that they have two copies of every chromosome. The reason that gamete cells have to be haploid is because when gamete cells merge with each other in the process of fertilization, each of them contributes one copy of the genome to the new developing organism. So when an egg is fertilized, that zygote that results is going to have one copy of the DNA from the mother in the egg and one copy of the DNA from the father in the sperm. And that's going to produce a diploid zygote cell, which will then go on through many processes of cell division to create somatic cells that will then become the lungs, the heart, the arms, the legs, all the various parts of that individual's body. So we're going to see that a cell will go through a cell cycle, which is essentially just a life cycle for the cell. And it's going to enter several different phases in this cycle, eventually resulting in division of the cell into two new cells. And then each of those two new cells would go through a cell cycle process of their own. So the longest phase of the cell cycle is called interphase, and really this just means the period of time in between cell divisions. In interphase, which takes up about 90% of the cell cycle, we see that there are three basic stages, G1, S, and G2. And in the G1 phase, the cell is growing. It's increasing its cytoplasm, proteins, organelles. So all of that stuff that's inside the cell and suspended in the cytoplasm is increasing in volume and number. In the S phase, we see that the cell is still growing, but the important thing here is that the DNA that's in the nucleus is going to go through a process of replication at this point. And we're going to look at this in just a moment. But the DNA replication process we'll talk about in a great deal of detail in Module 4. Essentially what's happening is every chromosome makes a copy of itself so that when that cell divides, every new cell has a copy of every chromosome. And then in the G2 phase, the cell is going to continue to grow until it reaches a point where it's ready to divide. Now, one thing that we want to point out is that during interphase, DNA is found as long fibers, and we call this chromatin. Those long fibers are associated with proteins, but it's kind of stretched out, and you can kind of think about it like yarn when it's unwound and it's just kind of flowing around everywhere, and it has the ability to get tangled if it's not organized well. But during interphase, that's not really a big deal. In the nucleus, that DNA is very accessible because it's not wound up tightly. And that's what we want, because the DNA is what we're going to use to build proteins that we need in the cell throughout the process of interphase. So the DNA is kind of what we call uncondensed. It's just stretched out everywhere. And we'll see that this has to change as we move into mitosis. The last note about interphase is that there is a stage called the G0 phase, or the resting phase, and some types of cells will enter this G0 phase where they essentially exit the cell cycle. And this resting phase could be permanent, or it could be a phase where the cell just kind of sits. It doesn't really grow, doesn't really do anything but perform daily functions, and then it will re-enter the cell cycle later. But in many cases, it's a permanent exiting of the cell cycle and will never go through the process of cell reproduction. Okay, after interphase, we see a period of time called mitosis, and this stage is actually very, very short compared to interphase, but this is where we're going to spend most of our focus in our discussion of the cell cycle, because mitosis is the division of the nucleus, and it's really important for us to understand how that genetic material gets divided up so that both cells get identical genetic information in the new daughter cells that are produced. And then the last stage is cytokinesis, which overlaps with mitosis. So very often we'll see that mitosis and cytokinesis are kind of discussed together at the end. But this is going to be where the cell itself divides. So as the nucleus is splitting up, we're also going to see that the cell itself gets split up at the end. And this is just another picture of the cell cycle showing essentially the same things that we just discussed. We have G1, S, and G2, and we see here that what's happening in each of these stages. Mitosis is divided up into 4 or 5 stages depending on which book you consult, and then cytokinesis is that overlapping stage where we actually see the cell split into 2 new cells. Okay, so let's take a quick moment to look at interphase. We're not going to spend a whole lot of time here, but if you recall, I said in interphase, we have G1, S, and G2 phases. So before the S phase of interphase, this is kind of what our cell looks like. And obviously there's all kinds of other stuff going on here. But we have in our nucleus, let's see, we have 4 chromosomes right now. And one thing we need to know about chromosomes is that they occur in pairs. And so these 2 chromosomes would have the same type of information. So maybe there's a gene right here for blonde hair, and on this gene or on this chromosome there's a gene for hair color, but maybe that's a gene for brown hair. Okay, so the same type of information, but it's not necessarily identical. And the same thing, these 2 chromosomes that are in a pair will have the same type of information, but it may not be identical. And so we call these homologous chromosomes, and these are homologous chromosomes. Homologous means same, and so we're looking at the same type of information. These chromosomes are not yet replicated because we have not gone through the process of S phase yet. The other thing I'd like to point out is, if you remember, one of the structures that we talked about in chapter 6 was the centrosome, which is our microtubule organizing center, and it plays a big role in mitosis and meiosis. So in animal cells, the centrosome is going to be a large organizing center with 2 centrioles that are organized at right angles to each other. And the centrosome is going to be replicated during S phase as well. So right now we see 1 centrosome located in the cytoplasm. However, after S phase, we now see that the centrosomes have been replicated and our chromosomes have been replicated. So whereas before S phase we only had 1 copy of the chromosomes, we now see that there are 2 copies of every one of these chromosomes, and they stay attached to each other they stay attached to each other, which is what gives us that X-like appearance that we very often see whenever chromosomes are represented. So before S phase, this is what our chromosomes look like. And then after S phase, they make a copy of themselves, and this is now what they look like. Okay? So, if we now move from interphase into the first stage of mitosis, which we call prophase, we're going to see that there are three major things that happen in prophase. And the first of these is that the chromatin that we talked about before has to condense into chromosomes. So, if you recall, we said that in interphase, our chromatin, our DNA, is kind of spread out everywhere. It's kind of like if you have a whole bunch of yarn that's just kind of laying around in a basket. In order for these chromosomes to be moved in mitosis, that chromatin is going to have to be organized so that the chromatin doesn't get tangled up and start causing knots and break and cause problems for the DNA. And so we're going to see that the chromatin condenses into these nice tightly packaged chromosomes that we see here. This is very similar to taking that yarn and rolling the yarn up into a ball so that it's easy for you to pull, one color of yarn out without getting it tangled up with all the others. Again, if we look back, and I'm going to switch back to the last slide for just a second. If we look back at these replicated chromosomes, okay, I'm showing here what they look like when they're condensed, but mostly because I want to be able to point out to you some of the vocabulary. Remember that we said that this and this would be homologous chromosomes because they contain the same type of information. However, the two copies of the chromosome in one package, in one chromosome, are called sister chromatids. And as long as they're connected to each other, they're still considered a single chromosome, but they are called sister chromatids. And they contain identical information. So remember we said there might be a gene for blonde hair right here. Well, because this is a copy of that chromosome, there's going to be a gene for blonde hair here as well. And we have a gene for brown hair here, and we'll have a gene for brown hair here as well. So these chromosomes are going to be still in a homologous pair, but the two that are connected to each other, that are copies of each other, are called sister chromatids and have identical information. So when we look together, or when we look back at prophase and what's happening here, we can very clearly see those sister chromatids and the fact that they're held together. They're kind of pinching together at what we call the centromere. Okay, so that's the region where we see it kind of narrow in on the chromosome. But the sister chromatids are held together by cohesions, which are types of proteins along the length of the chromosome. Let's see, we also see in prophase, in addition to the chromatin condensing, we see that the mitotic spindle forms. So again, if we look back, remember we saw these centrosomes that were replicated, and the centrosomes move to opposite ends of the cell, and they start extending microtubules, which are a type of protein. And those microtubules stretch out across the cell. Some of them are going to make attachments to the chromosomes, and some of them are just going to kind of stretch out across the cell. But we call this entire structure of the centrosome and those microtubules, we call that the spindle, or the mitotic spindle. And so that spindle is going to form in prophase as well. The last thing that happens in prophase is actually going to happen in what we call prometaphase in our textbook, but many people still consider this a part of prophase. I personally consider it a part of late prophase. But essentially, the thing that's going to happen that's a big deal here is that the nuclear envelope is going to break down. Now, when the nuclear envelope breaks down, that makes those chromosomes accessible. Okay, and so the microtubules from our spindle are going to start to form attachments at the chromosomes. So the major things that happen in prophase are, again, the chromosomes condense, the mitotic spindle is going to form, and the nuclear envelope breaks down, which is going to be the thing that allows that mitotic spindle to now reach out and grab those chromosomes. Excuse me. Okay, so as you can see in the diagram here, we see that some of these spindle fibers are attached to the kinetochores, which are proteins found at the centromeres of our chromosomes. And they're going to act kind of like puppet strings to move those chromosomes around to get them oriented for the next step. We can also see that some of these microtubules have spread out and are not necessarily forming connections with chromosomes, but they're still going to play a role in separating the cell and pulling apart these chromosomes. So, if we were to look here in prophase and prometaphase, again, we've got our spindle that has formed, and we've stretched out our mitotic spindle fibers, which are microtubules, and those fibers have started to make connections with our chromosomes, which is allowed because the nuclear envelope has broken down. Okay, so that's a summary of prophase and prometaphase. So now we're going to look at metaphase, and the stage of metaphase is actually a fairly simple phase. Essentially what's happening here is, remember, we have these microtubules from our spindle attached to the chromosome, and they're going to move those chromosomes around until they have lined up across the middle of the cell, which we can see in the diagram over here to the left. So those chromosomes are going to be nice and lined up evenly in the middle of the cell along a region that we call the metaphase plate, and that's really just an imaginary line across the middle of the cell. That's it. That's all that happens in metaphase. Those chromosomes line up at the metaphase plate. And so we can see that here, that our, leaving prophase, our chromosomes are going to start to be pulled until they're lined up across the middle of the cell. Okay? And now moving into anaphase, our sister chromatids are going to separate and actually become two separate chromosomes. So remember, we said that a replicated chromosome is going to be made up of two sister chromatids. And those two sister chromatids are going to be pulled apart, because if we look at our diagram here, look at our diagram here, and we've got one microtubule attached to this side of the chromosome and another microtubule attached to this side of the chromosome. When those microtubules start pulling in opposite directions, because essentially what's going to happen is that centrosome is going to start shortening those microtubules to reel them back in. When that happens, this chromosome, because it's being pulled in two different directions, is going to be pulled apart. And we can see in this diagram over here that the chromatids are being separated from each other. And now that they're separated, they are considered individual separate chromosomes. And they're going to be moving to the opposite ends of the cell, towards these centrosomes that are pulling them in. Okay? As this is happening, the cell gets a little bit longer, and this is in preparation for the next step where we're actually going to see the cell separate. So if we were to look at anaphase, this is us leaving metaphase. And remember, we had all of the chromosomes lined up across the middle of the cell. And now what's going to happen is those microtubules are going to shorten and pull the chromosomes in opposite directions. So the sister chromatids separate, and now as chromosomes, they move in opposite directions to opposite ends of the cell. And then in our final phase, telophase, we see that essentially we're reversing all the stuff that happened in prophase. And so we're going to see that the nucleus is going to reform around our chromosomes at each end of the cell. Our microtubules, our mitotic spindle, is going to break down. Each of those new cells is going to have a centrosome, because the centrosomes are already positioned at opposite ends of the cell. But those microtubules that the centrosome has kind of spread out and sent out throughout the rest of the cell is going to break down. And then finally, once our DNA is nice and tightly packaged inside of or safely packaged inside of a nucleus, the chromosomes are now going to start to kind of spread back out and decondense back into that kind of stringy chromatin that we talked about in the beginning. The reason for this is because now this cell is going to go through interphase, and in interphase we're going to need access to all of those genes on the chromosome to be able to perform our regular functions, and we can't access those genes if they're tightly wound up around proteins in the form of condensed chromosomes. Okay, so that's what happens in telophase. Now keep in mind that we said that this next step, cytokinesis, overlaps with the end of mitosis. So very often we see that telophase and cytokinesis are kind of grouped together as a single step, because really these steps are happening at around the same time. So cytokinesis is the process in which we're actually going to divide the cell and all the stuff that's inside of the cell. So essentially what we see is that in animal cells, and we can see that in the diagram to the right, The cell has elongated, as we said before. We've got all of these events taking place where the nucleus is reforming, the DNA is becoming decondensed. But we see that a cleavage furrow forms. And this is essentially where we have a band of proteins that wraps around the cell and squeezes in until we separate that cell into two new cells. Eventually, that cleavage furrow will completely separate these cells. But in animal cells, we have different challenges than what we see in plant cells. Okay, so in plant cells, we have to form a new cell wall, and we don't see that in animals. So plant cells do things a little bit differently. When the cell gets to the point of dividing and separating, we see that the cell is actually going to start to produce little vesicles that contain cell wall material, and it sends those vesicles to the middle of the cell, and those vesicles will fuse in order to form what we call a cell plate. Now, that cell plate is very close to a cell wall. However, it's not complete until the cell plate eventually grows and merges with the existing cell membrane and cell wall. So on this side of the cell plate you have membrane, and on this side of the cell plate you have membrane, and that's going to produce the new membranes for your cells, and then you'll have cell wall material in between them. So this is how plant cells handle that challenge of cytokinesis and developing a new cell wall between them. And so if we look at telophase, again, we can see here that our nuclear envelope reforms, which you can see is indicated by the dotted lines. Our spindle is completely broken down. We still have our centrosome, but the spindle is broken down. And these chromosomes, which are still shown as being fairly condensed in this diagram, are going to start to loosen up and become a little more stringy, like we talked about before. In cytokinesis, we're going to see that the cell pinches in the middle, and a cleavage furrow forms that will eventually allow those two cells to completely separate from each other. Final result, we now have two daughter cells that are identical to the original parent cell. So if you went back and looked at what the original parent cell looked like, you would see that it looks exactly like these two cells. And so now each of these cells will begin its journey through the cell cycle, going through interphase, and then eventually getting to the point where it can go through mitosis and cytokinesis as well to produce more cells. And this happens very, very rapidly. Relatively anyway, in organisms. It happens at different rates in different types of organisms, but it's important to note that this is how we grow. Okay, so we all start as a single fertilized egg, a zygote, and that zygote will divide so that we have two cells, and then those two cells will each divide so that we have four cells, and so on until you have a great big ball of cells that can now start to differentiate into different cell types and start to form structures. But mitosis is going to have to go on for a very, very long time, which is why it takes so long for a baby to actually develop, is because of all of the growth that has to occur to produce enough cells for that individual to be able to function properly and have all the parts that they need. Okay, so let's take a step kind of away from plant and animal cells, and let's look at what happens in a single-celled organism, like a prokaryotic cell. And we're going to be looking at an example of E. coli here, which is a type of bacteria. They go through a process called binary fission, which is a fairly simple process. And essentially what happens is that organism grows to about twice its size. It has a process of DNA replication that occurs, and so you can see here, if this is my cell, this is the DNA in the nucleoid region, and that DNA is a circular chromosome in prokaryotic cells. It has an origin of replication, which is where the replication process starts. It replicates or doubles its DNA, and now in this cell we have two copies of the DNA, and the cell has maybe gotten a little bit larger. And then that process of the replication process continues as the cell continues to get larger, and then when the replication process finishes, the two chromosomes separate from each other as the cell also is separating from each other, and we get two daughter cells as a result. So here We don't have a nucleus to deal with. We don't get the spindle and all those fibers. We don't have multiple chromosomes to worry about lining up. We just have a single chromosome that we double and put into 2 new cells. So it's a much simpler process than what we see in eukaryotic cells. Okay, we're going to wrap up with a short discussion about things that can go wrong because the cell cycle is a fairly complicated process in eukaryotic cells, and because of that, it has the potential to have a lot of problems. And so what we have in place in eukaryotic cells over time, we've seen the evolution of a regulation system or a cell cycle control system. So there are molecules in the cell that trigger and coordinate key events in the cell cycle and cause these events to stop so that we can check what has happened up to that point. And so your cell has these checkpoints where it does stop, and we check to make sure that everything has proceeded normally. So if we look at the diagram here, we can see, for example, after the G1 stage of interphase, the cell stops to make sure that growth has occurred properly and to make sure that any damaged DNA is identified, because that cell does not want to then go on to the S phase, go on to the S phase where it's going to double the DNA if that DNA is damaged. And so it would stop the cell cycle completely if it found damaged DNA. And then after the S phase, we go into the G2 phase, and before going into mitosis, it stops again. And here the cell is looking to see if there's any unreplicated or damaged DNA as a result of the S phase. So it's looking for any errors in DNA replication, and it's really just checking to make sure that that cell is really ready to go through division. And then in mitosis, before the end of mitosis, we see another stop or checkpoint, and this is going to be where the cell stops to make sure that chromosomes separated properly, that nothing's misaligned, and that it's okay for these 2 new cells to be released and start their own cell cycle. So these stop points or checkpoints occur in normal cells to make sure that the cell cycle has proceeded properly. If a problem is identified at one of these checkpoints, the cell is probably going to go through a process called apoptosis, or programmed cell death. And this is going to be in response to signals that are produced either as a result of stress or through one of the checkpoints. So the cell would recognize that something's wrong. It would signal to itself that apoptosis needs to occur, and it goes through this process of basically fragmenting itself and breaking down so that it is no longer a functional cell, and these parts can be phagocytized and recycled. Now, apoptosis is something that happens when something's wrong, but it is also a process that is very natural and very necessary for proper development. So in some cases, apoptosis must occur in a developmental pathway in order for that organism to develop properly. So for example, in the womb, we all have webbing between our fingers at some point or some stage in development. And when we're born, that webbing is gone. And it's because those cells in between our fingers have been eliminated through the process of apoptosis. So a lot of the modeling of an organism as it develops occurs through the process of apoptosis. Now, one of the things that can happen if a cell goes through those checkpoints and does not catch the problems, we can end up with cells that are cancerous. And essentially, a cancer is going to be a group of cells that grow faster than the cells around them, or uncontrolled, so they are not regulated properly. And so really, cancer is just all about cells that go through the cell cycle abnormally. And so in a cancer, we typically see that a single cell gets transformed. Usually your immune system catches these cells, or these cells are caught during the cell cycle checkpoints. But if a cell is able to kind of scoot through those the cracks in those systems, then it becomes cancerous and it starts to divide. And in many cases, it will draw a blood supply towards itself so that it can divide very, very quickly, and it divides uncontrolled or unregulated. Now, if Those cells do not have the ability to break off into the bloodstream or the lymphatic stream and spread to other locations in your body. We say that that tumor is benign. However, if those cells do have the ability to break off and spread, we say that it is malignant. And this is going to be the cancer that is most concerning. We call this process of spreading cancer cells to other parts of the body metastasis. And it usually is going to spread through the circulatory or lymphatic systems. And we'll talk a little bit more in Module 4 about some of the types of mutations that can occur in cells that will lead to the development of cancer, because these mutations in the DNA are going to be the things that cause the cells to now not be able to go through the cell cycle checkpoints like they're supposed to, and allow them to become unregulated. Okay, so that is it for Chapter 12. In Chapter 13, we'll be looking at meiosis, which, again, remember we said is similar to mitosis, but a little bit different. And this is going to be how your gamete cells go through the process of reproducing and becoming haploid.